we are going to talk about organelles in the cell today. And this is a lot of information. So just bear with me as we go through it. Maybe you've learned some of it before, and hopefully some of it's review for you. But let's start with the nucleus. So remember, we talked about yesterday how the nucleus, if you have a nucleus, it would be a eukaryotic cell. If a cell did not have a nucleus, it would be considered a prokaryotic cell. So here's our nucleus. Inside the nucleus, you have a smaller, dense structure called nucleolus. The nucleus has the DNA. That's where the DNA is. Sometimes you'll see the DNA as chromatin, okay, which just kind of looks like, I always describe it as like dental floss that's been like pulled out of the floss container, and it's just kind of a matted, stringy like structure that's like hanging out in the nucleus, okay? But when the cell goes to divide, then that chromatin condenses and comes together to form chromosomes, okay? So you only see chromosomes, they're only condensed to form chromosomes when the nucleus is about to undergo cell division, when the cell's about to replicate itself in the process of mitosis. Usually the DNA is going to be in the form of chromatin, okay? The DNA has got an envelope around it similar to like that of a cell membrane that's for its protection. And then there are pores on the surface of the nucleus, referred to as the nuclear pores, which are to allow things to go in and out of the cell. So again, this envelope is a lipid bilayer, just like the nucleus. And these pores are going to regulate things coming and going. Inside the nucleus, you also have in the membrane or the envelope, you also have a nuclear lamina, which is a series of proteins that helps maintain the shape of the nucleus. And we see that on the cell membrane as well, the extracellular matrix, that we'll talk about that as well. You don't see a lot of questions or anything about the nuclear lamina. Mostly I want you to know that the nucleus holds the DNA in the form of chromatin that condenses to form chromosomes during cellular replication. The nucleolus is in the middle. And, yep, that's all I need you to know. Okay? So, moving on. So, on our chromosomes, that's our DNA. Each eukaryotic organism has a specific number of chromosomes that they have. So for us, our chromosome number N equals 23. So that means we have 23 pairs of chromosomes. Sometimes you'll see it written as N equals 23 or 2N equals 46. So that's our, we'll talk about that more later, but our haploid number is 23. Our diploid number is 46. So whether you're talking about paired chromosomes or individual chromosomes. Chromatin is the DNA plus the proteins that helps coil up and to make it the chromosomes. And then the nucleolus, also you need to know about the nucleolus that that's where the ribosomes are made, okay? And the ribosomal RNA. And you should remember that, like we said previously, the ribosomes are important for protein synthesis. That's where proteins are made. So the fact that the nucleolus makes those makes it really important. Here's kind of our chromatin. This blue string here is the chromatin. Okay, and so you see it winding up here. This is going to form a leg or an arm, whatever you want to call it, of a chromosome. These are proteins. We'll talk about them more later, called histones, that the chromatin winds around to condense and form chromosomes. So again, here is just that matted dental floss looking stuff that's just kind of stringy and wispy in here before it coils up into the chromosome. There's your nucleus with your nuclear pores, your ribosomes studded on the surface of the nucleus because that's where they're made. And then continuous with the nucleus, we have what's called the rough ER. And we'll talk about that more in just a second. And then the nuclear pore that's going to allow things to come in and out. So those are going to be made up of, can anybody guess? proteins. Okay, just like those cellular membrane proteins from yesterday. So, ribosomes. We've talked about ribosomes. You're going to be sick of ribosomes because they do everything. They're like so important. Because they make proteins, and proteins do everything. They're so important. So think of proteins, think of ribosomes. So ribosomes are made up of rRNA and proteins, and they carry out protein synthesis. That's where proteins are made. So if you look at a ribosome, you have a large subunit of a ribosome and a small subunit of a ribosome. And they work together as a ribosome. You don't ever, well, you do see sometimes the large and the small subunits separated, but they work together. It's not going to work as a ribosome in protein synthesis unless you have both the small and the large subunit present. So ribosomes can exist as free ribosomes or bound ribosomes. I'm going to go back to this picture from yesterday, okay? So sometimes you will see ribosomes in the, like just in the cytoplasm, okay? I'm trying to highlight some of them, okay? Ribosomes just in the cytoplasm. But then also, those would be free ribosomes. Also, you'll see ribosomes that are going to be bound to the rough ER and to the nucleus, okay? And so that's the difference in free and bound ribosomes. Free ribosomes suspended in the cytosol or the cytoplasm, and bound attached to the rough ER or the nucleus. So the rough ER. So the ER stands for the endoplasmic reticulum. There's a rough ER and there's a smooth ER, depending on whether or not they have ribosomes. So the rough ER has ribosomes, the smooth ER has no ribosomes. The rough ER, like we said previously, is continuous with the nuclear envelope. You saw that in the picture up here. It just extends off of the nucleus, okay? And then the smooth ER is not continuous with the nuclear envelope in that same way. So the rough ER, it helps with proteins. It helps modify proteins. So proteins are made on the ribosomes. Remember, ribosomes can also be on the rough ER. But then after those proteins are made, sometimes they need modifications. So let's go back to another picture from yesterday that I'm thinking of. Whoops. Should still be able to name those 3 parts of a cell theory, hopefully. Okay, so here's a protein that has been modified with a carbohydrate side chain. So something like that addition to that protein would happen in the rough ER, okay? That's considered a glycoprotein because it has that carbohydrate on the surface of it. And so that happens in the rough ER, synthesizes glycoproteins and other secretory proteins. The smooth ER, what it's going to do is it makes lipids, okay? It also is important in detoxification. So one thing I want you to understand about organelles and cells is there's different cell types, and different cells have more and less of different things. So, for example, which organ in our body does detoxification? Does anybody know? You should say the liver, okay? The liver is responsible for a lot of the detox in the body, processing medicines, alcohol, all those types of things. And so the liver, cells of the liver are going to be very rich with smooth ER, okay? They're going to have lots of smooth ER that have a lot of function in detoxification, okay? And then also in lipid synthesis. Where is lipid synthesis important? To make the plasma membrane, to make the cell membrane, to make other lipids like cholesterol, okay? And so on and so forth, okay? And storing calcium ions. So there's the rough ER on the nuclear envelope, and I don't have a good picture of the smooth ER here. I apologize. Oh, here's the smooth ER. Okay, so the smooth ER looks a little different. It looks more like these tubes. Okay, it looks more like these tubes, but what you do notice is that it doesn't have those ribosomes studded on the surface of it. Okay. And then the next one is the Golgi apparatus. So the Golgi apparatus is kind of like a shipping and receiving center. So we consider our ER to be like a factory. I think that's what it said here, a biological factory, because it's really helping with synthesizing things, synthesizing lipids, synthesizing proteins, and so on and so forth. And then our Golgi is our shipping center. So once proteins are made, they may go through the Golgi and be shipped outside of the cell. So the Golgi is like the post office. So what you see about the Golgi is that it has two sides of it. The cis side is the receiving side. So that's going to be the side that's closest to the rough ER. So these two aren't connected, but they're close. Okay? And so once the proteins leave the rough ER, they are going to go in a transport vesicle to the Golgi apparatus, and they're going to come in on the cis side of the Golgi apparatus. And the Golgi is going to process them, okay? And they will package them, and they will send them outside of the cell. These transport vesicles will fuse with the plasma membrane and ship them outside of the cell on the trans side, okay, of the Golgi. So the cis side is the receiving, the trans side is the shipping, okay? So they are the packager of the proteins. Now, one other thing is important about proteins and how they're made and where they're made. So remember, we said ribosomes can be free-floating or bound. So ribosomes that are free-floating in the cytoplasm, those make simple proteins that typically stay inside the cell, okay? If it's a simple protein that doesn't need a lot of a modification and it's going to stay within the cell, then it would probably be made on a free-floating ribosome, okay? If it's a protein that's going to need a lot of modification, it's going to be made in the rough ER, because the rough ER has the capability to add those side chains and those carbohydrates and all those other different things. And it can send it then to the Golgi to be shipped out of the cell. Okay? So that's a big difference in the free-floating ribosomes and the bound ribosomes and the proteins that those make. Okay? Next is the lysosomes. So the lysosomes are important because they are digestion, okay? They get rid of things in the cell, clean up things that we don't need anymore. So obviously in the cell, things go wrong sometimes, and incorrect proteins are made, or things malfunction and need to be digested or destroyed. And so that would be the job of a lysosome. So they contain hydrolytic enzymes, which digest molecules and break things down. They can also digest food, sugars and other energy molecules and stuff for the cell. Lysosomes can do that as well. So that would be called phagocytosis, all right? And so we see that here in this first picture. We have this digestive enzymes in the lysosomes, and so here comes our food vacuole. A vacuole is just like a locker that holds stuff. So this is a food locker. It's kind of locker I like to have. And the lysosome binds with it. So we've got our lysosome bound to our food locker, and it pushes the digestive enzymes into the food vacuole and starts to eat them and breaks down those food molecules and digest. And so that process is called phagocytosis. Phago literally means eat, and then cyto, anything cyto is cell. So that means cell eating, phagocytosis. Another is autophagy, and so that's when we have a damaged organelle, okay? Again, you see that word phage. Phago is eating, auto is going to be eating like itself. And so we have something, maybe a mitochondria that's not working here, or whatever. And so the lysosome binds with this. It releases its digestive enzymes inside, and then it breaks it up into smaller parts, okay, and eats that. So a lysosome is for digestion, for breaking down things. Sometimes good food molecules, sometimes damaged organelles or things that need to be gotten rid of in the cell. Sometimes also like bacteria and viruses and stuff like that, they will be like targeted for destruction. And so that could be the job of like a lysosome as well. So vacuoles, here's our vacuole again. Vacuoles are for storage. So we got a couple of different types of vacuoles depending on what cell type we're looking at. In plants, only in plants, you have this large central vacuole, okay? And that just holds water generally and starches. And that helps a plant to remain, the word that we use is called turgid, which we'll talk about that more later. But if you think about the structure of a plant, you don't want your plants to be wilty. You want them to be like strong and upright and, full of water if you want them to be able to like stand upright. So in organisms, we're made a little bit different. We have a skeleton that kind of helps us to do that. Plants don't really have that skeleton, and so they depend on this water vacuole, this large central vacuole, to be full of water to help them stay upright and stay plump, so to speak. Food vacuoles, like we saw previously, that hold food, and then contractile vacuoles. Contractile vacuoles are important for helping pump out water. But what I want you to think of a vacuole as is a storage center or a locker, okay? Maybe. A mitochondria. This is an image of a mitochondria. Mitochondria is where cellular respiration happens. So cellular respiration, I'll just go ahead and write the formula down here. In case you don't know, you will know. So oxygen plus glucose yields carbon dioxide and water. That is the formula for cellular respiration, and that happens in the mitochondria. So remember, cellular respiration is the process that our body uses to take in glucose, which is what we eat, the sugar that we eat, and turn it into ATP, which is the sugar that our cell, or the energy, not sugar, the energy that our cell uses. And so that happens in cellular respiration is the process, which happens in the mitochondria. And we'll spend a whole unit on this. That'll be our next unit, actually. Actually, two units out. But it happens in the mitochondria. So when you look at the mitochondria has an inner membrane and an outer membrane. These folds in the mitochondrial wall are referred to as the cristae, and the inside of the inner membrane is called the matrix of the mitochondria, the mitochondrial matrix. And that's where cellular respiration is going to happen. The chloroplast. The chloroplast is only going to be in plants, okay? Now, chloroplast, if you look at the equation for photosynthesis, it is exactly opposite of the equation for cellular respiration. So plants are going to do photosynthesis. They also do cellular respiration. But they also do photosynthesis, and that happens in the chloroplast. So the chloroplast, if you see it under a microscope, it's going to look green because it has the pigment chlorophyll in it. Like the mitochondria, the chloroplast has two membranes. It's a double membrane structure. Inside the chloroplast is the stroma. That's kind of like the cytoplasm inside the from the cell. You got cytoplasm in the cell. The chloroplast has stroma inside of it. And then the other structures inside the chloroplast are these things that look like, I call them pancakes. So the individual pancake is called a thylakoid, if I can spell thylakoid. And then the stack of them is called grana. Okay? And that's where photosynthesis is going to happen within the chloroplast. So the thylakoid and the grana. The chloroplast also has its own ribosomes inside. Okay? And one other thing that's unique in the chloroplast, as well as in the mitochondria, is they both have, I don't know if it's drawn in the mitochondria here, you don't see it, but they both have their own set of DNA. The chloroplast and the mitochondria both have their own set of DNA, okay, which is different from the nuclear DNA. I said something wrong. I misspoke. I said that the the photosynthesis happens in the thylakoid. That's partly true. That's where the light is harvested. Part of photosynthesis does happen in the thylakoid, but that's where the green pigments are. So that's responsible for drawing in the light. So yeah, that's true. Photosystem 1 and 2 happen there. Sorry, it's been a year. So the endosymbiont theory. I mentioned this briefly yesterday when we talked about cell theory. I said all cells came from pre-existing cells, but where did the first cell come from? And so this goes back to endosymbiont theory. And so what happens in the endosymbiont theory is it kind of describes how we got eukaryotic cells. So we know prokaryotic cells are the older cells that would have been the first cells, would have been prokaryotic cells. And so how did we get these eukaryotic cells? And so that's what the endosymbiont theory kind of describes. And so we have this cell that's an ancestor of the eukaryotic cell. You see here there's already a nucleus, okay? So basically the thought here in the endosymbiont theory are that they engulf other organelles. So you had a prokaryotic cell that kind of engulfed another prokaryotic cell, and so that kind of became like your eukaryotic cell. And then you have this semi-eukaryotic cell, the ancestor of the eukaryotic cells, that has engulfed a non-photosynthetic prokaryote that becomes a mitochondrion. And then this engulfs a photosynthetic prokaryote, which becomes a chloroplast. And so this is what gives us our photosynthetic eukaryote that we have today. So we will talk about this again in more depth in evolution, but that is kind of the idea as to how we got from a prokaryote to a eukaryote. So I just wanted to give you a little preview of it today. Basically, you had a prokaryote that engulfed other components, other cellular-like components that formed our modern-day eukaryote that was capable of photosynthesis, that had mitochondria, that had chloroplast, and those types of things. Okay? Now, breaking from that back to our organelles, another organelle is a peroxisome. So peroxisomes are responsible for oxidation. So oxidation basically means taking hydrogen atoms off of things and pushing them to oxygen atoms, producing hydrogen peroxide. Okay? Now, these are going to be compartmentalized within the cell. Hydrogen peroxide is like toxic to cells. That's why if you've ever had like a nasty cut or something, sometimes they'll take and pour hydrogen peroxide on it and it'll bubble up and kill all the bacteria, and it kind of kills, those cells in that area, which they're on the surface, so it's not that big of a deal. Usually they're already kind of dead anyway. But it pulls hydrogen out of solution, so it can kind of help stabilize pH a little bit and produces hydrogen peroxide. Okay, and that is our last organelle for the day. Sorry that was kind of long. There's a lot of organelles to talk about. So I want the thing that's easiest for me and what you're going to work on today is to come up with a way to remember them. So you have an activity called a cell city analogy that's got like 9 or 10 organelles on it, maybe 11, I don't remember. And it's a little story, and each organelle has a job. It kind of has a function in the town. And so you're going to work through that one and identify the organelles there. And then I want you to write your own cell city analogy. Yours won't be a cell city. You could do something else. I've seen really cool ones, like some people will do like a car. A school is really common. That's like super boring. Don't be boring and do a school, but some of you will because you're boring. I mean, I love you anyway, but you know. I've seen some really cool ones though. Factories are pretty common. I can't think of any I've seen right off the top of my head, but come up with something really cool. Coolest one gets 5 bonus points on the test. I'm saying it now. Coolest one gets 5 bonus points on the test. Also, for anybody, whether you do a cool one or not, I will let you do extra organelles. So you're required to do the organelles that are already listed on the front on the cell city example. But if you do extra organelles in addition to those basic ones, maybe you add in a peroxisome, like that's not one that's on there, but maybe you add that in or add something else in, then you can get bonus points for each additional organelle that you add in. So you'll work on those today and use them to familiarize yourself with the cell. If you have any questions, let me know. You'll have a great day.